Hi everyone, welcome back. So in this video, we're going to be talking about special senses. Specifically, we're going to focus on the chemical special senses, so smell and taste, and also vision. All right, so first, just to kind of draw a distinction between your special senses and your general senses. So a lot of the general senses we've already talked about. So general senses are ones that are scattered all throughout the body, that are not localized in any one particular area. They are generally pretty simple in terms of their structure. So those general senses include touch and pain, temperature, proprioception. All of those are kind of general senses. Your special senses, on the other hand, are ones that are localized. So the structures are more complex and they tend to be localized in the head. And there are complex neural pathways associated with these that are much more specific than the ones associated with your general senses. So just to review some of the basics of the general sensation. So we talked about free nerve endings before. We talked about mechanical receptors that can feel pressure and touch vibration. We talked about muscle spindles that feel the stretch in your muscles. So those would all kind of fall under the general somatosensation. But now we're going to focus more on the special senses, which are much more specialized. And instead of being located all over the body, like you've got the ability to sense touch all over the body, you can only sense vision. You can only sense visual things through your eyes. You can only sense smell through your nose. So these special senses we're going to talk about are much more localized. All right, so we're going to start off with olfaction. So olfaction is the fancy word for smell. So we're not going to talk about olfaction totally on its own because olfaction and gustation are related. So gustation is another word for taste. So your sense of smell and your sense of taste are very closely interrelated. You really can't have one without the other. So your sense of smell is incredibly important to how things taste to you. So both of these things work together, and the pathways, the neural pathways that are associated with them, interact with the limbic system as well. So things that smell distinct to you, you generally form memories associated with those things. And smells can be very good at refreshing old memories that may have been kind of tucked away, because when you associate the smell with something, it's going to be associated with that memory. All right, so focusing in on olfaction here, we talked about the olfactory nerve being cranial nerve number 1, and how that olfactory nerve branches off the base of the brain, and then we have an olfactory tract here. So I'll highlight it in yellow. This olfactory tract here that comes underneath the brain and right at the top of the nasal cavity, and then it ends in this olfactory bulb. Right, so up here is a kind of zoomed in view of that. So olfactory tract ending in this olfactory bulb here. And that olfactory bulb sits right on top of what we refer to as the cribriform plate of the ethmoid bone. So remember, the ethmoid bone is being that bone that kind of forms the top of the nasal cavity. So this particular part of that ethmoid bone is called the cribriform plate. It's a thin layer of bone that has tons of tiny little holes in it. And those tiny little holes are places for olfactory nerves to go through the bone and into the actual nasal cavity. So this is kind of like your bridge between the brain and the nasal cavity itself. So right at the roof of the nasal cavity, you've got a layer of olfactory epithelium here. That olfactory epithelium is going to have supporting cells along with the actual neurons of olfaction. So when you smell something, when some sort of chemical enters your nasal passages, it's going to trigger those neurons to form action potentials. The action potentials are going to get sent up into the olfactory bulb, travel down the olfactory tract, and into the brain for you to interpret as smell. Okay, so olfaction, the pathways associated with it are what we refer to as being rapidly adapting. And that means that you usually only smell something for a short period of time before you kind of start to get used to it, and then you don't particularly smell it anymore. So you walk into a house and you smell like the smell of cookies or whatever someone's cooking. that smell's gonna hit you right when you walk in, and then your sensation into it is going to adapt pretty quickly. So over time, it's not gonna smell as strongly to you anymore. The smell is still there probably, so those chemicals are still in the air, but you just don't smell them anymore because those neurons that are associated with picking up those particular chemicals have gotten tired. So they don't fire at the same rate for very long. So rapidly adapting sense here. Um, Olfaction is obviously supported by cranial nerve 1, your olfactory, but there's also a component of the facial nerve, so cranial nerve 7. And part of that cranial nerve 7 lends some parasympathetic tone to the lacrimal glands. And this is why really strong smells can sort of make you tear up because it's associated with the parasympathetic tone of the lacrimal gland. All right, so moving on to gustation here. So gustation is another word for taste. Taste is much more simple than olfaction. So olfaction, you have the ability to smell thousands and thousands of different chemicals, very specific different chemicals. With taste, it's much more simple. You can taste 5 different tastes, and that's about it. So you can taste sweet, sour, bitter, salty, and umami, which is like a savory flavor. And everything else that you taste is really just combinations of those 5 things in different amounts, right? So sweet, sour, bitter, salty, and umami are really the only 5 things that you taste. Anything beyond that is going to be combinations of those augmented by your sense of smell. All right, so looking at the taste buds themselves. So you've got a whole bunch of taste buds located mostly on your tongue. The number of taste buds you have starts off high, and then they decrease with age. So the older you are, the less your ability is to be able to taste different things. Um, Taste buds themselves are composed of taste cells. So these taste cells are the ones that are actually picking up the chemical sensations that we associate with different tastes. All right, so if we look at this picture here, so a picture of your tongue here, on the surface of your tongue, you've got all those little bumps. Those little bumps are called lingual papillae. So lingual papillae are these little bumps on the surface of your tongue. Those are not taste buds. All right, so those are lingual papillae. If we take a look at, say, one of those lingual papillae and we do a cross section, so take a look at this picture here. We look at a section of that, we can see that the lingual papillae has these kind of little valleys in between them. And within those valleys, we find these little taste pores. And inside those taste pores are where we find the actual taste cells, right? And so those taste cells are the ones that are picking up the chemicals associated with different tastes. So if we look at it, within each taste pore, we could have all of these different taste cells. So even within a single taste pore, you could be tasting things that are bitter, salty, sweet, umami, and sour. So not every single taste pore is going to be just associated with one particular taste, although there is some evidence to support that, for example, you have more sweet receptors on the tip of your tongue versus on the back of your tongue. All right, so what are we actually tasting? If you taste something salty, then you're tasting the presence of sodium ions. That's all that is. Sour is the presence of hydrogen ions. So think of more hydrogen ions, the more acidic something is, the more sour that's going to taste to you. Sweet is the presence of monosaccharides, so just simple sugars. Bitter is usually tasting some alkaline compounds or some nitrogen containing compounds. And then umami is glutamate, so tasting the presence of glutamate compounds. All right, so how do we interpret these different tastes? Well, 3 cranial nerves are involved with the sensation of taste. So we mentioned these before in the previous video. So the facial nerve, cranial nerve 7, is facilitating the taste buds on the anterior 2/3 of the tongue. Glossopharyngeal, cranial nerve 9, is facilitating taste on the posterior 1/3. And then the vagus nerve also plays a role here because it's innervating the back of the throat. It's going to help assist the gag reflex. So if something makes you gag, it's triggering that vagus nerve. All right, so which one of the following are special senses and which ones are general senses? So look through these: touch, taste, equilibrium, pain, proprioception, temperature, pressure, smell, hearing. visceral, and vision. All right, so here's your answers. Touch is a general sense. Taste is a special sense. Equilibrium is special. Pain is general. Proprioception and temperature are general. Pressure is also general. You can feel it everywhere on your body. Smell and hearing are special senses. Those are located in the head. Visceral is a general sense, and then vision is special. Okay, so what's the main differences between the general senses and the special senses? What cranial nerve serves your sense of olfaction? Where are your taste buds located? And then what cranial nerves serve the gustatory sense? So here's your answer. General senses tend to be distributed widely across the body, whereas special senses are centralized, usually in the head. What cranial nerve serves as the olfactory sense? That would be cranial nerve number 1, the olfactory nerve. Your taste buds are located within taste pores among the lingual papillae, so along those valleys between the lingual papillae. And then which cranial nerves serve your gustatory sense? Those would be cranial nerves 7, 9, and 10. All right, so moving on to the eye here. So the eye is really your most complex sense. There's a lot of structure, a lot of physiology that controls how the eyeball works, how it looks. So we're going to go over these, starting with the accessory structures exterior to the eyeball itself. So we've talked about these extraocular muscles before, the fact that you've got 6 extraocular muscles that control movement of the eyeballs. So those are the superior, inferior, medial, and lateral rectus, and also the superior and inferior oblique. In addition to that, you've got muscles that are associated with the eyelids themselves. Another name for eyelid is palpebrae. So the palpebrae are your eyelids, and those are controlled by muscles as well. So cranial nerve 3, the oculomotor nerve, is the one that's controlling most of those extraocular muscles. So 4 out of the 6 are controlled by the oculomotor nerve, and the palpebrae are also controlled by the oculomotor nerve, or specifically levator palpebrae superioris is by cranial nerve 3. The other 2 extraocular muscles that are not controlled by the oculomotor nerve are the lateral rectus, which is going to be controlled by cranial nerve 6, which is your abducens, and then the superior oblique, which is controlled by the trochlear nerve, which is cranial nerve 6. I'm sorry, cranial nerve 4. All right, so let's look at some of the structures associated with the eyeball here. So we've got kind of a sagittal section through the eye. So note we can see the palpebrae here, so the palpebrae being the eyelids. The eyelids within them have a tarsal plate, and that tarsal plate is kind of like a tougher sheet of connective tissue, and that's because the skin of your eyelids is some of the thinnest skin in your body, so we need those tarsal plates in there to add a little bit of extra strength so that your eyelids don't tear. All right, so we've got the tarsal plates within the eyelids, and we've got eyelashes out here that are protecting, catching stuff that might fall into the eyeball itself. And then lining the inside of the eyelids and then lining the anterior surface of the eyeball are the conjunctiva. So conjunctiva are membranes that cover the inside of the eyelids and the anterior surface of the eyeball. So that's your conjunctiva. And then we can see the extraocular muscles here around the outside of the eye. So from this view, we can see the superior and inferior rectus and superior and inferior oblique. Okay. We should mention a little bit about tears. So you have lacrimal glands on the outer surface of either eyeball. So towards the lateral side of the head, you've got these lacrimal glands that sit kind of superior lateral to the eye itself. That's where you produce lacrimal fluid, and lacrimal fluid is just another name for tears. So the tears are produced in those lacrimal glands, and then they get washed across the surface of the eye, and then they drain into a nasolacrimal duct. So that at the interior corner of your eye, the medial corner, we then drain through this nasolacrimal duct and into the nasal cavity. So that's where your tears ultimate endpoint is in your nasal cavity, which is one of the reasons why when you're crying, your nose tends to run. It's because the extra lacrimal fluid you're producing is washing down into your nasal cavity. All right. So if your eyes are watery, a couple reasons for that. Either you're overproducing lacrimal fluid, so you're just making too much of it, or those nasolacrimal ducts are blocked and the fluid has nowhere to drain to. So in which case it would create a whole lot of extra fluid in the eye. All right, let's look at the anatomy of the eyeball itself. All right, so the eyeball itself has three different layers. Sometimes those layers are called tunics. So the outermost layer, the outermost tunic, is called the fibrous tunic. Fibrous tunic consists of the sclera, that's like the whites of your eyes, and also the cornea. And the cornea is the clear anterior surface of the eyeball. So that's the outermost layer. The middle layer is called the vascular tunic. That's made of the choroid, which is a kind of a dark layer that's highly vascularized, and also the iris. And then the innermost layer is the nervous tunic, and that's your retina. All right, so if we look at this picture of the eye here, we can see all three of those layers. So here the fibrous tunic is represented in blue, so we can see this the sclera on the outside of the eye, and then the cornea on the anterior surface of the eye. So that's all fibrous tunic. Then we've got the vascular tunic, which is the choroid. So choroid here, and then anteriorly we have the iris. And then the innermost layer is the retina, which is your nervous tunic. So that's where the actual nerve cells are. Okay. Um, so of the three parts of the middle tunic, or your vascular tunic, you've got this, the choroid, but in addition to that, we've also got the iris and a muscle called the ciliary body. So ciliary body are these tiny little muscles that are going to attach to little strings, and those little strings are going to pull on the lens of your eye to help change the shape of the lens so that you can focus near and far away. I'll show you a picture. All right, so here we can see the choroid and we can see the retina, or I'm sorry, we can see the iris. Right, so iris here on either side, and then just behind that we've got the ciliary structures here. So we've got the ciliary muscles attached to these suspensory ligaments. And again, over here, ciliary muscle attached to suspensory ligaments. And those suspensory ligaments are holding on to the lens of the eye, and they will be able to either pull on it or push on it, change the shape of that lens. And as the lens changes shape, it's going to be able to focus either further away or closer as we change the shape of it. All right, and then the innermost layer is that nervous tunic, which is the retina essentially. So the retina has, in addition to just having the nerve cells, there's also epithelial cells that are pigmented with melanin. And this is really important that the inside of the retina here is pigmented with melanin. And that's because you're bringing a lot of light into your eyes, and we don't want the light to just be bouncing around everywhere in there. So the melanin that's on the inside of the eye is there to absorb light. So it's not constantly scattering all over the place. Otherwise, everything would be super bright all the time. So we need that melanin so that you don't have to constantly squint your eyes. Okay. Now the exact center of the retina, so this is what your eye doctor looks at when he looks inside your eyes. The exact center of the retina is called the macula lutea. So macula lutea is the center of the retina. And then right in the middle of this macula lutea is the fovea centralis. So the fovea is like the center of the retina. Um, Now, in this picture here, in addition to the macula lutea and the fovea centralis, we see this other structure off to the side here called the optic disc. Optic disc is not at the center of the retina, and in fact, the optic disc has very poor vision in comparison to the macula lutea, where we have the sharpest vision of your eye. So the sharpest vision, the highest concentration of nerve cells associated with photoreception are right in that macula lutea, fovea centralis. Right? The optic disc that I pointed out in the last picture is actually your blind spot. So the optic disc has no photoreceptors at all. You can't see anything. So light that hits the optic disc is completely invisible to you just because you do not have the photoreceptors there to pick up that light. So if we look at this picture here, all along the side here, we've got photoreceptors. So these are your photoreceptors, these little tiny cells here that are picking up light signals and then transducing that into action potential and sending that along this optic nerve here. So this is the optic nerve. sending all that information along the optic nerve to your brain to be interpreted as vision. But right where that optic nerve enters to the back of the eyeball, there are no rods and cones there. There's no nerve cells there to pick up any light sensation. So your optic disc is where the optic nerve enters, or I should say leaves the eyeball. And where it leaves the eyeball is your blind spot. Okay, so which cranial nerve controls 4 out of the 6 ocular muscles, including, and in addition to that, the levator palpebrae superioris? What glands produce tears, and what do we call tears technically? And then what drains the excess tears from the eyes? All right, so the cranial nerve that controls 4 out of 6 extraocular muscles plus levator palpebrae is cranial nerve 3, the oculomotor nerve. The glands that produce tears are your lacrimal glands. Tears are called lacrimal fluid. And then draining the excess tears from the eyes are the nasolacrimal ducts. Okay, your sclera and your cornea belong to what layer of the eyeball? What layer is the iris in? What tunic contains a layer of melanin producing epithelium? What is the optic disc? And which cranial nerve is your optic nerve? All right, so there's your answers. The sclera and the cornea are both part of the fibrous tunic, so that outermost tunic. The iris belongs to the vascular tunic. The tunic that contains melanin producing epithelia is your nervous tunic. The optic disc is where the optic nerve exits the eye, and that optic nerve is cranial nerve 2. All right, so getting a little bit more in depth here into the actual cells that are capable of receiving light, so photoreceptor cells. Your photoreceptor cells are rods and cones. So they're named rods and cones basically just after the shape of the cell itself. So these outer segments of the cell were thought to look like, in this one here, thought to look like a rod, and this one here thought to look like a cone. And so they got their names by the shape of them, essentially. So rods are specialized to pick up low light images, and basically all rods can do is determine light from dark. So your rods allow you to see in black and white. Cones are specialized to pick up highlight, clear color images. So rods are more low light, so you can see in the dark by using rods. Cones, you need bright light in order to be able to see color. All right, and we've got 3 different types of cones. So whereas you just have one type of rod, there are 3 different types of cones, the red, green, and blue cones that are each going to interpret different wavelengths of light to allow you to see and interpret different colors. All right, so here's where these different cones work. So your blue cones are picking up colors around the 450 wavelength range. Your green cones are around the 530s. And then your red cones are getting up closer to 600. Okay. As far as blindness goes, so different types of color blindness, there are a few different types. They usually have to do with not having one particular type of cone. So red-green color blindness is the most common type of color blindness. And that's when you're missing either the red or the green cones entirely. So if you're missing them, you're not going to be able to interpret colors of those wavelengths. Night blindness, which is when people have a hard time seeing at night, is usually a problem with your rods. And rods, in order to produce their specific pigment that they use, which is rhodopsin, they need a lot of vitamin A. So vitamin A deficiency will cause a lack of the production of that rhodopsin, which causes night blindness. All right, a little bit more of the anatomy of the eyeball here. So here again, we're looking at the eye. We can see the cornea. So this is the cornea, that transparent outer layer, anterior surface of the eye. And then we've got the lens here, right? And the lens is this clear structure that's there to bend the light as it comes through the eyeball. And then behind the lens, we've got this whole region back here, right? And that's the kind of inside surface of the eye. So the cornea and the lens are both transparent so that we can bring light through that so it can hit the retina on the back of the eyeball. All right, so we can see, looking at this same picture, we can see these different areas of the eyeball. So the area, the large kind of gooey area in the middle of the eye is referred to as the vitreous chamber. So that vitreous chamber is full of like a thick gel-like fluid called vitreous humor. And then this anterior part of the eyeball here. So all of this from the lens on forward we consider the anterior cavity, and the anterior cavity is filled with aqueous humor, so kind of like a thinner fluid. All right, so that aqueous humor is going to be super important to nourishing those structures of the eye. So think of aqueous humor as kind of like a filtered version of blood. So it's got oxygen, it's got nutrients necessary to support those structures of the eye. And then it's constantly being cleaned and refreshed to keep that part of the eye from waste from accumulating in that part of the eye. Um, we also need the correct amount of humors in the eyeball, so aqueous humor and vitreous humor, so that we keep the eyeball itself inflated. The eyeball is basically just a fluid-filled bag. We need to have the right amount of fluid in there to maintain the right intraocular pressure, right? So we need the right amount of fluid to maintain the IOP. Right? So if we have too much fluid, for example, we have an increase of pressure on the inside of the eye, and that results in glaucoma. So glaucoma is an increased intraocular pressure. And if we have increased pressure there, you can imagine that there isn't a whole lot of space for that pressure to be relieved, so we can start to do damage, and you could start to lose vision because of that. All right, real quick about the pupillary response. So your pupillary reflex is that when you go into bright light situations, your pupil, which is just the hole in the middle of the iris, so the pupil isn't a structure itself, it's like an absence of a structure. The hole in the iris is your pupil. As you approach bright light situations, the muscles of your iris will contract so that that size of that pupil gets smaller, right? Because if we're in bright light, we don't need to take as much light in. And if we're in dark light, so everyone shuts off the lights, it's nighttime, whatever, the pupil will widen to take in the maximum amount of light in order to be able to see in those low light situations. So, and this is just a reflex of the muscles of the iris. So you've got two muscles of the iris that are contracting alternately to either expand the pupil or constrict the pupil according to how much light is in the environment. All right. As far as being able to see clearly, so many people are nearsighted. Another word for nearsightedness is myopia. Myopia means that you can see things up close pretty clearly, but you can't see things that are far away very clearly. And it basically has to do with the shape of the lens of your eye and where that is refracting the light coming in. So refracting just means bending the light. So over here in this image here, so nearsighted eye, we can see light coming in. It's hitting that lens, and then the lens is bending the light, and where all those rays of light come together is not hitting the retina, right? So the retina here is represented in yellow in this image. We're not hitting the retina, so that means that what we're looking at is not going to appear clear. So in order to correct that, you add a corrective lens, and that corrective lens could be eyeglasses or contact lenses, but you create another lens on the outside of the eye to change change the angle of the refraction of the light, so it actually goes all the way back and it hits the retina here so that we can send that information to the brain. All right, and hyperopia is the opposite. So this is farsightedness, where you can see distant things very clearly, but you have a hard time seeing things up close clearly. And it just means, in this case, the rays of light that are coming through the eyeball are being refracted by the lens, and the angle at which they're hitting is beyond the retina, so it's past the retina. So in this case, again, you would need corrective lenses in order to correct that and refocus those light beams so that they land on the retina itself. All right. So once the light hits the retina, so if we've got vision working pretty well, our lenses are refracting the light where they need to be refracting, or we're wearing corrective lenses outside of that. That means that the light that we're interpreting is going to come in, hit the retina, and then that visual information is going to be funneled towards the optic nerves. So you've got one optic nerve coming out of each eyeball here. Those optic nerves then go back in your head and cross over right underneath the inferior surface of the brain there. They're going to cross over. Where they cross over and form that X is called the optic chiasm. After they form that X, then they're going to migrate all the way to the back of the brain. So this information is going to travel along these optic tracts all the way to the occipital lobes, and the occipital lobes is where your primary visual cortex is. So way in the back of the brain is where you're going to be interpreting those light signals that are coming in through your eyes. Okay, so a few more questions here. What photoreceptors detect low light and develop low resolution black and white images? What photoreceptors detect color or bright light? What photoreceptors are concentrated around the periphery of the retina? And where is the primary visual area of the brain? All right, so answers there. The low light black and white images are rods. The photoreceptors that can detect color are cones. The photoreceptors that are concentrated around the periphery are the rods. So in fact, your peripheral vision tends to be kind of, if I'm looking at something just out of the side of my eye, I can't distinguish color that well outside in that periphery. It's because my rods are what's picking that up. And then the primary visual area of your brain is the primary visual cortex and your occipital lobes. All right, so I hope this was helpful. Let me know if you have any questions.